Excuse me. All right, so we are going to be taking a look at cell transport. Again, cell transport is this idea that we get stuff into and out of the cell. Remember that cell membrane, or sometimes called the plasma membrane, or sometimes called the bilipid or the bilipid the phospholipid bilayer, is selectively permeable, and it helps the cell to maintain homeostasis because it lets certain things in and it only lets certain things out. So, again, the focus of today, the essential question that we're trying to answer is, how does the cell separate itself from its environment and still allow materials to enter and leave the cell in order to maintain homeostasis? Again, let's remember that homeostasis is this idea, and by the way, it's also one of the characteristics of all living things, where we are able to maintain a balanced or constant internal environment, an internal environment which is in equilibrium. So, in order to do that, the cells help to maintain homeostasis, and we do so that how do we do so is the question that we are trying to answer today. All right, so hopefully you have your note catchers on. All right, so cell membrane. A semi-permeable membrane that surrounds the cell. So we are on part A, number one. The semi-permeable membrane that surrounds the cell and protects the cell's components from the environment surrounding the cell. So the cell membrane basically separates the cell from its outside environment. Okay. So when we talk about anything that is intracellular, we are talking about the space inside the cell. The prefix intra means inside. Um, when we are talking about spaces that are extracellular, we are talking about spaces outside of the cell membrane. And please remember, all cells have a cell membrane. Again, it does not matter if it is prokaryotic without a nucleus. It doesn't matter if it's eukaryotic with a nucleus. It doesn't matter if it's a plant cell, a fungal cell, an animal cell. It doesn't matter. All cells have cell membranes. Side note, all cells also have DNA. All cells also have ribosomes, and all cells also have cytoplasm, excuse me. So those are the four things that all cells do have: are cell membranes, DNA, cytoplasm, and ribosomes. So every single cell on the planet has those four things. All right, moving forward. This is part two of notes under cell membrane.
I got a question.
Oh, yes, please go ahead.
Which one is this? Like, is this like a new assignment or this is the one we've been doing?
So this is, I just posted this one today. It should be under 11 16 number 2. I literally just posted it this morning.
So basically, this is the new handout, right?
Yes, yes, it is.
So that one that we just did was the first one.
Correct.
Okay, because I was really confused. Thank you so much. I appreciate it.
Yeah, yeah, no problem at all. All right, so this was again cell membrane, and again, all cells have cell membranes. Cell membranes surround the cell to keep the insides in and the outsides out. This is number 2 under cell membrane, maintaining homeostasis. So, what is the function of the cell membrane? The function of the cell membrane is to maintain homeostasis within the cell. Remember, and we've talked about this ad nauseum, homeostasis is a stable internal environment. Homeostasis means that there is equilibrium inside that living organism. Without being able to maintain homeostasis, organisms can die. It doesn't matter if it's a single cell, it doesn't matter if it's a human being, it doesn't matter if it's a tree. If you cannot maintain homeostasis, you cannot stay alive. It is a requirement for life to maintain homeostasis. So cell membrane, super important. It helps to maintain homeostasis, which means that's going to help keep you alive. Oh, everything alive. So, how does it do this? One of the things that it does to help maintain homeostasis is it's able to communicate with other cells. It can protect and support the cell itself. And it is probably the most important, selectively permeable. And selectively permeable means it regulates what goes in and what exits the cell. Whenever I think of selectively permeable, whenever I think of allowing certain things in and allowing certain things out, I always think of like those spy movies where you have the people and they're coming in to like the FBI or like CIA headquarters, right? And then like they always have to stop at the front desk. And like there's like the security guards, and you have to go through the metal detector and all that kind of good stuff. And then if there's a problem, they stop them. They're like, no, you can't come in. And then there's like the fight scene that happens in the movie, right? So that guard station is selectively permeable. It regulates what enters into the building and what can leave the building, right? So that's kind of the function of a cell membrane. So if you need a good analogy for your vocabulary, hint, hint, nudge, nudge, wink, wink, elbow, elbow. So basically, a cell membrane is like a guard station at guarding the front of the building. It regulates what goes in. It regulates what goes out. All right. So this is sort of a cell membrane diagram. I believe you have the same exact diagram on your handout. I'm going to leave this up for a minute just because this is kind of, the thing. Let me turn my pen on. All right. So the whole thing. Oops, come on. Why is this not working? The whole thing is called the phospholipid bilayer. Okay. That is like the chemical makeup of the cell membrane. All right. So in that phospholipid bilayer, we have the phosphate, which is hydrophilic. Remember, that means it loves water. And it has the fatty acids, which is hydrophobic. So each one of those little like things in there is a phospholipid. The round phosphate head is polar. The fatty acid is nonpolar. All right, so inside this phospholipid bilayer, because it is polar, um, it's going to let some things just slide right through, and we'll talk about that in a minute. But some things can't slide just right through between the phospholipids. They might be a little bit too big. They might be polar themselves. In that case, we need a couple of other things. We have the protein pump, and like the name suggests, it can help. It's protein, and it helps to pump things in or pump things out. We have the protein channel, and just like the name suggests, it's made of protein, and it is a channel. So it's kind of um like a tunnel, right? So things can kind of just move through there, just like a tunnel. We have the aquaporin, all right, and we have the glycoprotein. Oh, and we have some cholesterol here, but that's okay. All right. So these are the main parts of the cell membrane, which is kind of crazy because it's like one little organelle has all of this going on. But again, this cell membrane is super important to like survival of all living things because again, it controls homeostasis. It controls the ability to maintain a nice, level, balanced, in equilibrium internal environment, which is required for all living things. All right, moving on. Quick little video for. I'm sorry, go ahead.
I said, which ones are we writing down for that one?
Um, anything that okay, so again, the most important ones are the phospholipid bilayer,
Hmm.
protein channel,
Okay.
protein pump, and then knowing the parts of the phospholipid. So it's the phosphate head and then the fatty acid tail. The glycoprotein, the cholesterol, the aquaporin, those are nice to know, but they're not required.
Thank you.
Yep, no problem. All right, so our friends, the amoeba sisters, because we love them.
In my second year of teaching, I was looking to improve an osmosis lab I had done the year previously. Osmosis, if you remember from our osmosis video, involves water traveling through a semi permeable membrane, potentially a cell membrane. And I wanted a cool way that students could model this in different scenarios. And one of my colleagues told me about this egg lab. I won't get into the whole lab though. It was actually one of the first steps of the procedure that got to me. To get ready for the lab, my colleague told me, you can soak eggs in vinegar for 24 to 48 hours and the shell comes off. Oh, so I need to make hard boiled eggs then. No, raw eggs. But if the shell comes off, well, that's the whole point. What's underneath the shell is going to mimic a cell membrane. It's kind of modeling how a cell membrane would function. You know, if the whole egg was actually like a cell. So then you run different scenarios with it for your osmosis lab because it will be a semi permeable membrane, like a cell membrane. I couldn't visualize this. If the shell comes off, but it's a raw egg, how does it stay together? So I have this area in my house that's designated for things for me to try out. Don't worry, I always clean up afterwards. I tried this experiment out in advance, just to be sure. The hard shell is removed, but the membrane that was always there remains. We often visualize the membrane of a cell this way, like this membrane around the chicken egg. The cell membrane is semi permeable, meaning it lets some materials through, but not others. We have an entire video all about cell transport and how materials can pass through the membrane. A cell could never be as large as a single chicken egg, though. Why? Well, it turns out surface area is a very important thing. Remember that surface area determines the surface measurements of that cell membrane. The cell membrane controls what goes in and out of cells. That includes food coming
Sorry.
a very important thing. Remember that surface area determines the surface measurements of that cell membrane. And the cell membrane controls what goes in and out of cells. That includes food coming in, as well as molecules that are essential for metabolic processes, and then also waste going out. If volume, which is all this space inside, increases, then you will have more need for surface area, as you will have more need for food to enter, more of a need for waste to be removed, and more metabolic reactions occurring in this larger volume in the first place. If we do a little bit of math here between these two models, I'm going to use popular cube models instead of egg-shaped models because it's a little faster for me to do surface area and volume calculations. See how here there is a big difference in surface area to volume in the smaller model? 6-to-1 ratio. That means the surface area in this small model is 6 times more than the volume. Look at this beautiful ratio with so much surface area. But if we look at this bigger cube and we do some math for this model, that surface area to volume ratio decreases. Sure, the surface area is still larger than the volume in the large model, but it's only 2 times as large now, not 6 times as large. Cells are way smaller than this small model here to allow for an exceptionally large surface area to volume ratio. And a major reason why we're not going to find a cell as big as this chicken egg here. Surface area is important. And while we can model a lot of the processes of cell transport from this egg membrane and how important the membrane is, I don't want to neglect talking about the amazing cell membrane structure itself. Because the cell membrane structure truly is magnificent. And since every single living thing is made up of one or more cells, which is part of the cell theory, it's a big deal because every single cell has a membrane. It doesn't matter whether you're talking about bacteria or protists or plants or animals or fungi. Even archaea aren't too cool to have a membrane. They all have a cell membrane. The structure can vary some, but we're going to be talking about some major structures of the membrane that you can actually find in most cells. We should mention that the fluid mosaic model is often what we use to describe the cell membrane. A mosaic, in case you've ever created one, we did in some of our art classes over time, arranges many small pieces together to make some large piece. You'll see that that makes sense when describing the membrane in a minute. The word fluid implies movement, and this is true for the cell membrane as the components are floating around, they're not static. So let's take a look at some of these components in the cell membrane. We're looking first at a phospholipid bilayer. A phospholipid is a lipid, but an interesting one. So when you talk about a lipid in general, many lipids are nonpolar. Think of oil, for example. It's nonpolar. It won't dissolve in water. Water is polar. But a phospholipid is interesting because one part of it is polar, the head, and the other part of it is nonpolar, the tail. It's amphiphilic. Let's explain what we mean. We often refer to the polar head of the phospholipid as hydrophilic, which means that part loves water. Well, if a lipid could love. The nonpolar tails are hydrophobic. They do not like water. These phospholipids arrange themselves into a phospholipid bilayer with the nonpolar areas here in between, away from any water. It also allows this area in between to be separated from the outside and inside. Water can be found on the inside and outside areas. Also, these phospholipids, they don't just stay put. They move around. It's the fluid mosaic model after all. This gives the cell membrane flexibility. Phospholipids can even flip-flop around, but that's far less common. Remember that this entire phospholipid bilayer borders the whole cell. It would be a sphere, even though we're just looking at one area of it. We have an entire video that talks about which molecules can go in and out of this membrane and which ones can't that you can view, but for now, we're going to look at some of the other structures more in depth. Cholesterol. Cholesterol often gets a bad reputation. And while cholesterol that builds up in the arteries can be a problem, cholesterol in your cell membrane is critical. If temperatures drop, the cholesterol can actually function kind of like spacers between these phospholipids, keeping them from becoming too packed. Or vice versa, the cholesterol can actually function to connect phospholipids, to keep them from becoming too fluid in warm temperatures. Proteins. In protein synthesis, we talk about why it's so important for cells to make proteins. Many proteins are found in or on the cell membrane, and they play major roles. Peripheral proteins, like the name suggests, tend to be on the peripheral area of the membrane. So while they're on exterior areas of the membrane, they generally are not going to go through the membrane. That's for integral proteins. Integral proteins go through the membrane. Oh, and peripheral proteins can sit on them sometimes. Because of location, these proteins tend to have different functions. Integral proteins, with their potential to go through the membrane, are frequently involved in all kinds of transporting methods for all kinds of materials. Some relevance? Consider the breakfast you ate this morning. Your body digested what you ate for breakfast to obtain glucose. Once in the bloodstream, those glucose molecules can't just squeeze through the phospholipid bilayer to enter all of your cells. The glucose molecules are too big and polar. But your cells need glucose to survive, to make ATP, and they rely on integral proteins to get it. Peripheral proteins tend to be more loosely attached, since they're generally not stuck in the membrane. They have an assortment of functions such as acting as enzymes to speed up reactions or attaching to cytoskeleton structures to help with cell shape. Both protein types can have carbohydrates bound to them, which can make them considered a glycoprotein. If the carbohydrates attach to a phospholipid, you could have what's called a glycolipid. Glycoproteins and glycolipids can identify the cell as belonging to the organism, self and non-self recognition, which is very important when you're fighting pathogens. They can also be involved in many kinds of cell killing. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against many viral and bacterial diseases. Well, that's it for the mucus sisters remind you to stay curious.
All right, so that was a whole lot about the cell membrane, but we'll talk a little bit more about it as we go through. I just love the amoeba sisters, and I think you guys know that. Totally like their biggest fan. All right, cool. So we are now on part B, something called the concentration gradient. So the concentration gradient, this is part one, occurs when there is a difference between concentration of any substance on either side of the membrane. So concentration is how much is there, right? So looking at this picture, which side of the cell membrane has the higher concentration of molecules? Is it the extracellular, the outside of the cell, or the intracellular, the inside of the cell? What do you guys think? Where do we see the higher concentration of molecules, ladies and gentlemen? I heard somebody unmute, but I couldn't hear what you said. Which side of the cell membrane has a higher concentration of molecules? Extracellular, outside of the cell, or intracellular, inside of the cell?
Extracellular.
Extracellular, yeah, because you can see there's like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. There's 13 molecules on the outside, and there's only 4 on the inside. So there is a difference in concentration. Concentration is how much is there, right? So that is a problem, right? There is a difference in concentration. So let's see how to solve that problem. Now, there is looking at the concentration gradient, there are 3 possibilities. There can either be a higher concentration in the extracellular area than in the intracellular area. So maybe there's a higher concentration outside than inside. We see that in the first diagram. Second diagram, we see a lower concentration outside of the cell membrane than inside of the cell membrane. And in the third diagram, we see a concentration that is equal on both sides of the cell membrane. When you have a concentration that is equal in the two areas, then we say that that is in equilibrium. So that word equilibrium is what you need to add on that last part of the note catcher there. Okay, so when you have a difference in concentration, not equilibrium, but when you have a difference in your concentration gradient, you have an area of high concentration and you have an area of low concentration, molecules will naturally move from high to low. Sometimes we call that down the concentration gradient. And again, I always think of this as like water flowing down a hill. Like if you've ever been in the mountains, if you've ever been hiking, maybe Tennessee into Appalachia, the Rockies, anywhere at all like that. If you've ever, or even if you've just seen it on TV, we see these mountain streams and they always flow downhill. That is a good analogy for a concentration gradient. Water naturally flows from high altitudes to the low altitudes. Well, concentration gradients naturally go from areas of high concentration to areas of low concentration. That happens naturally. Naturally, molecules want to move down the concentration gradient. So, if you are moving down the concentration gradient, in other words, if you're going from high concentration to low concentration, this requires no energy. But if you are moving against the concentration gradient, if you are trying to go from low to high, it requires energy. Okay? And this is, again, my favorite analogy is about water. So, water naturally will flow from the top of the mountain, down the mountain, to the rivers and the seas, right? Now, does that mean that we can't pump water up to the top of the mountain if somebody like decides that they want to put in, like, I don't know, like a cabin or something? Of course we can. Of course, we can pump water up there, but we're going to have to run that pump. And that pump is going to require either electricity or gasoline or something. We are going to have some kind of energy pumping that pump to make the water go uphill. We can do it, it just requires more energy. So, again, Concentration gradient, molecules naturally flow from high to low, high concentration to low concentration, or what we call down the concentration gradient. That does not require any energy. You want to reverse it, that requires energy. All right, so this is where cell transport comes in. We are now in letter C of our note catcher. In order to maintain homeostasis, cells must transport materials between the intracellular, inside the cell, and the extracellular, outside of the cell spaces. Things have to move across that cell membrane. Because again, cell transport is going to help maintain homeostasis. It's going to be able to take in molecules that we need for cell processes. Maybe those are nutrients, maybe those are other molecules, maybe it's water. And if there's waste inside of the cell, remember we talked about the lysosome, it's sort of being the janitor of the cell, cleaning things up. Then the lysosome has to be able to get rid of that waste material. So it has to go across the cell membrane. So again, cell transport is super important. But depending on which way that concentration of waste or nutrients or whatever is going, is going to tell us what type of cell transport we have. Passive transport is movement of material across a cell membrane without using energy. In other words, whatever materials we're using are moving down the concentration gradient. In other words, they're going from high areas of concentration to areas of low concentration. This happens naturally. It does not require any energy from the cell. There are three types of passive transport. There is diffusion, there is facilitated diffusion, and then there is osmosis. So those three types of passive transport do not require any energy because, again, we are moving materials from areas of high concentration to areas of low concentration, and that flow happens naturally. So let's start with this idea of diffusion. This is part E in your note catcher. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. Molecules will move directly through a membrane. These are going to be non-polar molecules. These tend to be, a lot of times, they tend to be gases and water. Well, I shouldn't say water. These tend to be gases. So, like oxygen can diffuse this across the cell membrane, carbon dioxide can diffuse across the cell membrane. Gases do this a lot. And again, the driving force behind this is any movement of substances across the cell membrane. Molecules will just kind of randomly move in and out of the cell until equilibrium has been achieved. Again, equilibrium is all about the concentration outside and the concentration inside being the same. So again, diffusion, it is a form of passive transport. Does not require energy. Now, we have facilitated diffusion. It's still diffusion, but these are molecules that cannot move directly across the cell membrane, and they have to go through protein channels. Okay? These tend to be larger molecules. Okay? That's about it. So it works the same way. We are going from areas of high concentration to areas of low concentration. It doesn't require any energy. It's just that these are pretty large molecules, so they can't just push through the bilipid, the phospholipid bilayer. They have to go through a protein channel. In other words, they have to kind of like travel through a tunnel, if you will. Again, it is a form of passive transport. It does not require any energy. All right. We are going to stop the notes here for today.